Hello, my name is Manolis Maragkakis, and I'm a Studman investigator in the National Institute on Aging. Today, I would like to talk to you about recent work from my group to use direct RNA sequencing to capture the full-length transcriptome of cells upon stress. So, as you probably know, the ability of cells to respond to insults and stress decreases with aging, and particularly in aging-associated diseases such as neurodegeneration. So we're very interested to understand the regulation of the RNA state upon cellular stress that remains poorly characterized. Part of the reason why it's poorly characterized is because previous studies have either used short-read sequencing, thus missing long-range interactions on the mRNA molecules, or they used artificial reporters, which obviously lack in generalization to the full transcriptome. So in this work, our experimental setting is quite simple. So we have control cells and we have cells that we treat with arsenite. And then we perform long-read sequencing with direct RNA sequencing with the technical modifications of ligating adapters of the 5' end of the molecules, which essentially act as markers so that we know that we sequenced all the way to the 5' end at single nucleotide resolution. Here we see just a quick volcano plot for these data, where we see that indeed we see many of the genes that we expected to be upregulated upon stress, and this is just a validation of our experimental setting. But perhaps the most interesting thing is that now that we have these long-read sequencing data, we can dive in and look at features on the mRNAs that we could not previously see. And that here, for example, we look at the RNA length. And surprisingly, what we see is that upon oxidative stress, RNAs in cells become substantially shorter compared to control conditions. And this shortening, when we look at the meta plot, primarily occurs at the 5' end and not so much at the 3' end, indicating that there is a decay process happening at the 5' end. Now, we've seen this plot, but we also wanted to approach this in a more statistical way and be able to capture robust changes and changes that in transcripts that show robust changes across different replicates. So we developed Nanoplane, which you can think of as an equivalent of a differential expression program, but in this case, we're doing differential RNA length. So initially, we used simulated data, and we at different expression levels and different proportions of shortening, and we actually see that the software performs quite well and even controls quite well for the false positive rate. But most importantly, when we apply it on the real data, we see that indeed the transcripts that are being called by the software as the most significantly short are indeed the shortest ones. And we went into the process of validating some of the top targets, such as ASCCS3, using qPCR and designing primers at varying positions along the mRNA in order to capture the difference in expression that these different primers would report for the different parts of the RNA. And indeed, you see that the closest to the 5' end the reporters are being designed to, the less of the expression they report upon stress, indicating that this is capturing the 5' to 3' end decay as the Nanoplane software predicts. Now, we also wanted to dive into more on the molecular players for this stress-induced decay, and we found that XRN1, which is a primary exonuclease in cells, is indeed one of the major players here. And when we silence XRN1, we actually see that we have a very we have a rescue of the RNA length, but also that the RNAs, the transcripts that have been identified by Nanoplane as the most significantly shortened ones, are the ones that show the greatest rescue of their lengths, both validating the role of XRN1, but also it validates the Nanoplane software itself. Interestingly, we see no association between decay at the 5' end and deadenylation. So contrary to what the traditional decay model under normal condition would predict, which would be that the first step, it would be deadenylation and then subsequent decapping and fragmentation from the 5' end. That's not what we see upon stress. Interestingly, what we saw, though, is that these RNAs that are being shortened the most are those that have the shorter coding sequence and the lowest GC content. And we hypothesized that maybe translation might be playing a role. So we use different inhibitors of the stress response, or we use cyclohexamide to force to freeze ribosomes on the mRNAs and prevent ribosome runoff. In both cases, we see that we can rescue the stress-induced decay, indicating that translation is actually protective, and translation inhibition is a requirement for the progression of this decay upon stress. Quite interestingly, though, what we also found is that these RNA fragments that are being created upon stress actually persist in cells and are not immediately cleared. So we don't see a correlation between 5' end fragmentation and RNA abundance. And this made us think that perhaps this indicates that there is a storage mechanism. There's an active mechanism in the cells for the storage of these RNAs. So we looked at the role of the possible role of the stress granules in this process, and we indeed found that the stress granule transcriptome is enriched in transcripts that show increased shortening, indicating that these membraneless organelles might be playing a role in the storage of these decaying RNA fragments, perhaps until the stress is resolved, so that the cells can then deal with these RNAs after the cells gets resolved. So thank you for your attention, and I look forward to discussing with you further.